I'm Nicholas Cole and I'm the group leader of this laboratory, and my laboratory uses zebrafish to investigate the causes of motor neuron disease to perhaps search for a cure of motor neuron disease. And we're also interested in how limbs develop, and we wish to understand which genes and proteins make limbs form in the developing embryo. We use zebrafish in my research because it has many key advantages which complement other research models. Firstly, the zebrafish genome, the proteins and genes that make the zebrafish form and function, are very similar to those in humans and other vertebrates. Also, we can obtain lots and lots of embryos very easily from the zebrafish. They are fertilized externally, so when the lights come on in the morning we can get up to 300 eggs from each female fish. This gives us lots of material to work with, and there are also some very interesting technologies which allow us to overexpress genes in the zebrafish. So for example, in our motor neuron disease project, we are able to overexpress human proteins in the fish, and we know that these proteins carry mutations in the patients which cause motor neuron disease, and by expressing these same proteins in the fish we can hopefully understand motor neuron disease and search for a cure. Because we're able to get so many eggs and larvae from the zebrafish, this is a considerable advantage to us. So we are able to get lots of eggs and larvae from the motor neuron disease carrying zebrafish, and we can divide these eggs up and treat each group of these eggs with a separate drug or compound, and hopefully this will lead to some understanding and potentially a cure for motor neuron disease. In my laboratory we have this fish facility. We have this relatively small room which has these fish racks in it which are full of different types of zebrafish. So each of these tanks contains a separate strain of fish, which is a completely different experiment. This is a really efficient way to use animal models. If you could perhaps imagine this was a facility with mice in it, it would be enormous and require a large number of people to run it. But this is a fairly compact model system which we're able to run relatively cheaply and with very few people, and yet gives us very efficient research outputs. Our other specialised equipment includes microscopes, so we have microscopes with fluorescent lamps on them, and this allows us to see different fluorophores, so very bright red, green, blue marker proteins. So in real time in a living fish we're able to tag a protein with a fluorescent marker, and this allows us to see what that protein does, when it's made and where it goes with a fluorescent marker. So for example, we have fish which express green, blue and red fluorescence in different proteins all in the same fish at the same time while the fish is alive. So not only do we have our own microscopes, but we make extensive use of the confocal microscopy and advanced microscopy that's available through the Bosch Institute, and we also make many of our fluorophores and constructs using the Bosch molecular biology facility. A typical day in the lab would involve collecting eggs and injecting those eggs with our constructs to make them express the proteins and genes we're interested in. And we would also spend our time analysing our fish. So once we've got the fish to express the normal and the mutated genes, for example, we can then analyse the whole swimming performance of the fish. Do they locomote normally? Do the neurons fire normally? For example, we can look at motor neurons and neuromuscular junctions and investigate whether or not we have in fact got a motor neuron disease phenotype. By this I mean, do the fish show signs of motor neuron disease? At the moment we have very little understanding of why motor neurons die in motor neuron disease, despite the fact that we know of several genes which are mutated in patients which cause motor neuron disease. So the main goal of my laboratory is to try and understand this process and this mechanism in motor neuron disease, and we hope in some small way this will bring us closer to a cure.